Okay, then I would suggest that we start. Well, I hope you can already enjoy the few memes I share here in my first slide. Throughout my presentation, I will share some of them. If you appreciate, I just created a stream on Image Flip, so if you want to go find more of these memes, share yours, uh, you can go to that link. Okay. Um, but maybe going into the presentation. So my name is Tom Boissonnet. I work in the Center for Advanced Imaging in Heinrich University in Düsseldorf, Germany. I work also for the NFDI for Bioimage, which is a consortia of different universities working on data management. Um, and today I wanted to tell you about the value of annotated data with OMERO. And I seen that for this halfway to I2K, there's no other introduction to OMERO. So I will also cover some of the introduction to OMERO and take this side of why annotating your data can be useful. Um, and since we're talking about data management, I think we could not escape talking about FAIR, but I also wanted to make it slightly different way than usually because I think a lot of us have heard about FAIR before, so trying to change a bit things. And so I presented in this Venn diagram having, well, the four terms for the FAIR: findable, accessible, interoperable, and reproducible. And so if we're finding ourselves at the center, that's when we would be doing FAIR. But what about the other corner? So if I remove the findable from FAIR, getting this AIR acronym, I would be kind of like a dragon sitting on my pile of gold. So I created a great data set that's annotated, well understandable for everyone that you could access, but nobody knows where it's really located. So I'm kind of trying to hide this treasure from others. Um, we could also look from the without accessibility. So maybe I'm a captain who has a treasure, but I will not tell anyone. Well, everybody knows about it, but nobody knows how to access it, basically. Or I could do it without the interoperability. So maybe there's an alien coming and wanting to tell me about the secret of the universe, but I cannot understand whatever he's saying. Or maybe for reproducibility, where I will be this alchemist that by accident found how to create the philosopher's stone, but forgot to take his notes, so there's no reproducibility. So I thought it could be fun, yeah, presenting FAIR in that way, that uh, what are the different words really meaning if we remove them from FAIR? Okay. And if I look at the two on top, that's more when we would be talking about data publication. So if I'm making my data sets findable and accessible, that would be something I would handle rather at the moment I deposit data. But for what's reproducible or interoperable, that's something we can already work on in the lab. So data publication, if you had to go for one place for your microscopy data, I could only recommend you to try depositing this in the bioimage archive. They also accept metadata, provided that you still have this metadata and you know how to find it. But that would be the right place for microscopy data. But if I look more from the perspective of doing data management in the lab, that it's not just about publishing your data, making your data fair is kind of the goal we want to achieve as a community, that other people sharing their data set, I could reuse them. But I want to also take the side that yourself in your lab should use data management, because that's also going to benefit you in your daily daily lab routines. And so what I mean by data management is for everyone. By everyone, I mean the group leaders, the experimenters, or the bioimage analyst. And I distinguish these three roles, but nothing stops someone from having more than one of these roles. So you could very well have an experimenter that also does his own image analysis, or a group leader that sometimes goes in the lab and does some experiments. But what is the interest for these different people with data management? So the group leaders will need to have this kind of overview of projects, want to know what's going on in the lab, want to be able to monitor the progress of the different student, but also it's about the long-term archiving, that once a student is gone, that they would have the possibility to find back this data and understand what this data is. Looking on the side of an experimenter, those are normally the people who know all the details about the projects, about an experiment. So you know if there's some problems that occur during the experiment, if some had an issue, and so they can organize and annotate the data. They are the one creating the data, and they have the role of also explaining their colleague what is in this data set. And in third, for the bioimage analyst, they will need the metadata for processing. So there's different forms of metadata, but they will need to have access to this information. And they will do processing of the data, but then they will need to share the results back. So not just that they need to get the data to process, but they will also need a way to share the results back with their colleague. And so we need to have a kind of traceability of an analysis. So if I find some outliers as a result of an image analysis, that I would be able to trace it back to what part of the image actually was showing something odd, for example. Okay. So there are these different motivations to do data management in the lab. But I would also like to state that because we're doing image data management, this is about microscopy, everyone needs an easy way to see the images. Okay. So if you have questions, don't hesitate putting them in the chat as well. So I want this session more as a demonstration, so it's not really a hands-on session, but if you have questions, please feel free to write them in the chat and I will try to take them as we go. Okay, and talking about scientific imaging, microscopes are primarily measuring devices. And we write images. We have a different dimension for our images. So you're all familiar, I hope, with microscopy, that we can acquire image in different channels. Those would be an image splitted across the 3 channels, and I could make a composite image, so overlaying the channels and mapping them to some colors, so that I would have this colored image of my cells. But just the image only are not enough. I would also need to add some metadata to the image to make it complete. And so images also come with metadata. And the metadata I'm talking about here is the acquisition parameters, so the metadata recorded by the microscope. So that would be all the microscope, the lens used by the microscope, so the magnification or laser power or all this kind of information the microscope has access to. In the metadata, we can also talk about the different fluorophore we use. So that would be here saying for these channels that we use certain fluorophores, and displaying also in this image the scale bar, so that without this extra metadata, our image cannot really be used for scientific experiments. And there's more dimension to those. So the Z slice or the T dimension. And just to show you some examples here. So this is the same image that was actually a Z stack. So I'm also displaying it at the different position in the stack. Or images can also be time series, in which case we want to see changes across time in our field of view. But microscopy image are quite complicated. And I want to make here a distinction between an image and a file. So you are already probably familiar with this kind of mapping that from a single file you have a single image behind. That's very common to have. What's special about microscopy, that can range from a few megabytes to several terabytes if we're talking about very large data. So something like volume EM, where you would acquire very, very, very large cubes of data, or SPIM images, so something like light sheet that would acquire a lot of this stack for a lot of time points, or high content screening that would acquire thousands of images across different wells with different conditions. But we have this other kind of mapping, so we could very well have in a single file several images. That would be the kind of zip archive that, for example, Leica provides. Or we could also have several files for a single image. So if we are splitting our images into different files, so one per plane. And that kind of makes it uninteresting. So that's not the kind of problem I want to have to handle in the lab. So I don't want to be doing file management. Again, we want to do image data management. So we want an easy way to see the images. And I don't want to have to deal with this kind of multi files for single image things, etc. This is already a problem that has been addressed. So how do I read the file to open an image? And you probably already know Fiji, and that is very famous for that you can take a single file, drag and drop on Fiji, and that will open an image. But that's actually not really Fiji operating behind the scene. You might have seen this window before. And looking closer, this is bioformats. So bioformat is a library that's coming by default with Fiji, and that's the thing that's going to read your images. But bioformat does not only read the image, it can also read metadata. So if you tick these boxes, you will be able to also read microscope metadata from your image files. And bioformats does support a lot of formats. So here is the list of formats supported that you could find on their website. But there's actually a few more pages that keeps on going. And so currently bioformats support this 160 file formats that you could read, for example, with Fiji now. Okay, so now let's dive a bit into OMERO. So OMERO, you might have seen before. We're gonna go and take some time. I will present you OMERO and features, but you have this web interface into which you can organize your images. This is what we are calling OMERO Web, so the part that we are interacting with in our browsers. And this connects to another OMERO component, that's the OMERO server, so something you don't really have to worry about, but it's on the server that you would be uploading your images. And again, here, it's not going to be OMERO server reading your data directly. It's coming with bioformats. And bioformats will then take care of reading the pixels in your image, but also reading the metadata of your microscopes. And to give you the full story, then the people who made bioformats and the people who made OMERO are the same people, so the OME team from Dundee. Okay. I still see no questions, so I will just keep going. And yeah, please don't hesitate again if you have a question to put them in the chat. So what you are seeing now is the OMERO web interface. So these are data sets that come from our production server of Heinrich Heine University. I am not showing you research data. I am showing you data that we acquired with a master module. So that's data sets that students acquired and that we made them upload to OMERO so that they could also practice with OMERO. So trying to teach the student early on about data management. So what you see in this interface, you have on the left the list of your data sets, images, and those, whatever you select, you're going to have thumbnails in the center to give you a preview of the image you have in this data set. And on the right, you're going to have annotations and information about the thing you're selecting. So right now, selecting a data set, what it will show me is the name of this data set if I wanted to rename it, also the data set ID, who owns the data, and some other description that I could work on to describe my data set if I wanted to. And the first thing I like to show my users about OMERO is that now that you're using a web client, what you can use to share your data is not sending files anymore. You don't need to send files to someone anymore. What you can do is find a link to that resource. And if I was sending this link to someone else working in this OMERO server, so someone in that group, they would be able to see the same resource. And so I could point them to a data set, or I could point them to a single image or to other things that I can show you later. So the first nice thing about this link, well, that you can share the data with someone else, but that's also something that you could start using in conjunction with your electronic lab notebooks, for example. So say that you're writing down in your e-labs an entry about the experiment. You went to the microscope with 3 samples, and and for these 3 samples, you acquired sets of images. You could simply take the link of your data set and put them in your e-lab. And the other way around, you could also write down in the description of your data sets, you could write down in the description of your data sets the link of the electronic lab notebook entry. And so that from both directions, you would be able to find the data sets that you generated. So find your data sets from the eLab, or find the notes you took about your experiments from the data. Looking more inside the metadata that is coming from the microscopy image. So selecting this image and going to this acquisition tab we have in OMERO. This is where it would display me either the original metadata. So that corresponded to the original metadata option in bioformats in Fiji. I don't know if you remember these 2 tick boxes. And that would really show me all the metadata that exists in the file, really as a list of metadata. And this list of metadata is not formatted, so it's really given row by bioformat. And the reason for that is, well, every microscope file format writes metadata in their own way. And here we have a Zeiss file, so that would be the Zeiss layout of metadata. But what a bioformat does is to also try to interpret this metadata into and transform it to the OME model, which is a metadata model for microscopy, something open that they try to that would try to harmonize how we write microscope metadata. And instead of looking at the original metadata, I could then look at the transform metadata into this OME model and find then what is the name of my objective, what is the model of my objective, what is the magnification of my objective, or if I would go for the different channels, what were the excitation, emission wavelength, fluor that was used in that experiment. So OMERO is capable of showing me this metadata coming from the microscope. Going back to this general tab, we can also see that there's already some metadata information given for any images. So that would be on the dimension of an image or pixel sizes. So again, something that is read from the microscope metadata. And here we have channel names. So that is not always something that OMERO is capable of extracting from metadata. So sometimes it works, sometimes it does not. That's going to depend on how your microscope writes metadata. But that is also something that you would be able to change here and name your channels. And that would be the first kind of metadata we would write ourselves in OMERO. So making sure that all our images for their channel names have the right channel name. And that will be useful for us later in this session. We also have other kind of annotation we can work on. And I want to come back to that a bit later, but not to just leave you confused here. We have these tags that are a way to assign categories to our images. So indicating or sorting images or data sets could also be tags. Well, most of the objects in OMERO could be tagged, or having this key value pair annotation that would be describing in detail the content of my image. And again, that's something I want to come back on a bit later in this session. Okay, so I still don't see any question in the chat. I will pause for a minute just to drink. Okay. So continuing with the basis of OMERO, this is the window where I can view all my data, have the overview of the different images I possess in my group. But exists also in OMERO a viewer. So if I take, for example, this image and I'm gonna double click, this is gonna open the viewer, and the viewer does exactly as the name suggests, is to view your data. So here you don't need to find the files, drag and drop in Fiji anymore. You would already have a viewer integrated in OMERO that you could browse your data, and that would be here for a Z-stack, going through the different Z-planes, have an idea of what's in the data. But you could also play here directly with the channels, so toggling on or off the different channels, that I would be able to adjust the contrast, the intensities for the different channels. Because of course, overlaying all channels together might not always be easy to see the different data, so that you could turn them on or off. You also have the possibility to change the color mapping, so the lookup table for the channels here, or a bunch of different options. The nice thing here about OMERO is that you could save your settings, but those are really just display settings. So once you upload an image in OMERO, the image would stay in its raw format and would not be edited by my actions in the OMERO interface. What you would be saving, the annotations or the settings, is really something that is not modifying the raw data, but just adding an information or saving those settings for the image. And something else I like to show about this this viewer is that you could also open maybe the same image several times. So in these different panels, like so. And now what I am able to do is to log their views together. So with this log button here, I would be able to synchronize them. So moving inside one of the channels or moving inside one Z-plane, that would also update the other images. And what that's useful for is that I could now keep a single channel for every image, like so, so that I would be able to browse my image, my different channels, but in separated views, so I'm not overcrowding the views. And of course, better change the mappings to grayscales, for example, so that the intensity would be kind of nice for our human eyes. So I see a question from that. Is the saved setting image specific, or it could be applied to other images? So there's a way you can apply settings to multiple images. So save to all here would apply all the settings to the different images in this data set. So right now I selected a data set, so it shows me the 3 images of that data set. Another place you can do that is also here from the preview image, and updating the settings. Why is the view like that? No. Sorry, I don't know why I don't see that by default, but oops. In this preview, you can also adjust the settings where you also have the save to all button. And doing that in the preview can be sometime more efficient if you want to save the settings for objects like full screen. Well, I'm jumping a bit ahead of myself, but if I want to edit the settings for a whole 96 well plate, for example, so instead of changing the settings of the images one by one, I could update the settings for this plate image, save to all, and that would apply the setting for the whole plate. Okay. So I think, yeah, that's most of the things I wanted to say so far about the image viewer. And at this point, we are not really making use of metadata other than our channel names here that we could here already quickly identify what channel we are working with. So better than try calling them channel 1, 2, 3, and 4, that I would already have that information here. And a way to copy settings, I don't know of a way that's, sorry, so there's another question from Ved. So is there a way to copy paste settings to images in desired folders? I don't know a way from the OMERO web interface to pass it like that. There are ways programmatically to do it with Python or command line, but that would be out of scope for this. Or the one trick I would have for you, so you could also take the images with the settings you have, you could move it to a different data set to then apply the settings to all images in that data set. Then you would need to put this image back into the original data set. That's a bit of a workaround, but if you really need to copy your settings and doing it quickly, that's probably the workaround I would have. Okay. All right. So the next thing I want to show you in OMERO is the plugin called OMERO Figure. So the plugin figure, we can find it in different places. That also depends on your implementation of OMERO. But I could, for example, select this image and with a right click, go to open with OMERO figure. Another way I would have, so clicking on the button here on top, that would by default propose me either to create a new figure or to open a figure that I created before. I'm gonna do create a new file, and this is asking me for an image ID. So what I can do here is go find my image, and I could take, I could take the link to that image. I do Control C to copy and pasting it here, and OMERO will understand that this is the image it has to load. So adding the image, this adds the image to the canvas here in OMERO figure. And well, like the name suggests, again, this tool is used to create figures for your microscopy data. I also do not intend to do a full introduction to OMERO figure in this session, but I will nonetheless demonstrate what it can do, and again demonstrate why annotation can be useful here. But if you want more detailed OMERO figure introduction, there was recording, other recording for the I2K made. So if you search on Google, on YouTube for OMERO figure, I'm sure you could find more tutorials. But what I'm doing here, we have an A4 page with that image that I'm placing here. So I want to do a figure where I'm gonna show the different channels separately and make one of the image with all channels overlaid. And I need to roughly fit that into my A4 page for the simple reason that I don't need to export a figure that would be gigantic or fitting for a poster. So just trying to fit my image into this A4 page. So you can see I have the 4 panels next to each other. We have in OMERO figures some buttons to align those panels. So um, these small features that can really help you making well, straight figures. And so in the first panel, I'm gonna select all channels together. Let's zoom in a bit. That I want to here show really what's my image is looking like. In the other panels, I will again select a single channel for each. Yes, and so I'm now displaying my 4 channels next to each other. What I really need to do here, so displaying in my figure, if I wanted to show the DAPI channel here, blue on black is never really great for the human eye. So in terms of contrast, we don't really see the different gradients of blue. So it will always be necessary that when you are showing single individual channels, you will have to change their mapping at least to this black and white. There are, again, more lookup tables, so talking about things like these mappings that would also display a greater range of intensity using colors. But for today, we'll just limit myself to white. So I'm going to quickly change the mappings of these different channels, select all the panels together, and I'm going to display this stack first. And I'm going to copy and paste this row so that I would then show different layers of my Z stack like that. And I can again come and adjust my contrast intensities. So I would go with this cursor to adjust the intensity. What I would have to be careful here about are well, not to saturate the image. So you have also this button here to display the image in this high-low mode. So where pixel saturating from the top would be displayed in red, and pixel saturating from the bottom would be in blue. I cannot fix the blue color, so that's already the minimum value. But at least I should be careful that I'm not saturating my image here. And so trying to find settings that are a good compromise between not saturating and still having good contrast. And I see a question from Sergey. For a series of typical non-high content screening experiment, X image per condition, 3 for condition in each experiment, what do you recommend to encode as dataset, as projects, tags, key value? Thanks. So that I want to cover a bit later. Yes, how do we organize our data sets that at the end we have a kind of easy and coherent way to think about organizing data. So that I will cover slightly later. Okay, Okay, back to my figure. These are my colors. So let's finish this figure here. I'm gonna switch back to the normal mode. Now going to this label tab. This is used to display information on my image. So features about region of interest or inset panels. But what we should show, of course, is a scale bar. I could also fine tune the scale bar. So if I want the bar to be slightly less thick, probably displaying the label of 10 micrometer on each of this panel is not necessary. So let's remove it from here. And now what I'm going to have to write is what are the image showing? So putting some legends here. So for this image, I could, for example, position that label in the top left corner, writing it in white, and I could call that composite, for example. 12 is slightly too large, so let's go back to 10. And for my individual channels here, I could very well start typing the names for each of those images, but I would have to make sure that these images are the one with the DAPI and go back to type DAPI here. But since we already have that information, that we already took the time writing down the names of our different channels, this would be the first place where metadata becomes useful for you. So let's go and select all 4 images here, go to labels, and instead of typing myself, I have a drop-down list right here for which I could ask to display the channels in separate labels. Uh, let's put them for 10 in the top left corner of the image, and I could display them right away. So what happens here is that for every panel, it would look at what are the active channels and display the name of those channels in the panel. So if I would do the same for my composite image, now I could also add the information that's also going to be color encoded. And here I don't really have a recommendation or what would be the best way to do between writing composite or the name of the channels in the different colors. Probably there would be writing something like that and instead using the colors for the different channels. Here I'm not really certain of what to do. But that's maybe also the time I could point you to other materials. So from this I2K conference, there was also the talk from Helena Jambor on how to create figures or what do you have to consider to create figures. So that was a session that was last Monday. So I'm sure that's also going to be part of the session recorded and that's going to be published on YouTube later. She also has a publication about how to create figures and a checklist on what to do, what not to do when you're creating them. So I could only advise you to go check that. Okay. And so this is how to display information about the channel. So some metadata that I've taken the time writing on my images. And we will come back to figure a bit later. Okay. So I think now we could start talking about how do we annotate data and how do we organize data. Let's reset the zooming here. Okay. So what I'm going to tell you right now, it's not something that's inherently encoded in OMERO. So OMERO provides you a framework on giving you possibilities to organize data, possibilities to annotate data. But they did not make the choice for the users on how they should organize their data set or how they should annotate their images, etc. So I'm going to give you my own point of view and how I do my own organization, so you have something to adapt. Yes, but just so you know that it's not the only way that you would have the only way that you would have to do this. So how I like to think my data sets is that data sets are one replicate of an experiment. So in the example of Sergey saying that you have X images per condition and 3, 4 condition in each experiment. If you go to the microscope with 3 samples, at the end of the session, you should have 3 data sets. So you took X images for each condition or each X image for every sample, sorry. Organizing them like this, that you do obtain one data set with multiple image only for this sample. This will simplify my life later when I want to annotate, because I'm gonna have, I will want to describe my data. So I want to explain what is the condition of my sample, what is special about it, or did I do some treatment, or what are the different channels that I have in my image? And instead of writing that information on the image, I could write that down on a data set. And if I write this on the data set and I look at an image below, I would then have still that information displayed in OMERO web. So it would tell me these annotations are coming from the data set. And here I'm really explaining this sample had this sample ID, and this was condition A. If I started mixing different condition in a data set, so all the images I took on Monday, all conditions together, then I could not describe my data set anymore. I would have to describe every single image. And again, even if you were acquiring different samples in the same condition, the same would still apply, that even though you could describe most of the information as the same, you would still not be able to describe from which sample came every image. And for me, it's very different to do 6 image in one sample or doing 3 image in 2 samples with the exact same condition. These are still a different idea of replicates. So I would really, at the end of the day, if I go with 3 sample, end up with 3 data sets. And what you can see here in my organization, okay, that's getting too zoomed in. With my organization here, what I do is that for every data set, I start by writing the date of the data set and then some name. So the date is really important here, that I would, every time I'm adding a new experiment to my list of data set, they always end up or add at the bottom. So finding a data set becomes kind of easy. I always have an idea of when I was recording my data, so I could find them kind of by the date. There's different variation of that. If you have experiment IDs, and that's your way of thinking how you organize your experiment, you should go ahead and use your experiment IDs. But what's really important here, in my opinion, is to be consistent on your naming, that you're not mixing up different style and things are not appearing ordered. And then in the second part where I write the name, this is really where I would put something for me as a human to understand, so that I would have a kind of description that I can easily recognize. Okay. So that's the first part of your question, I think, Sergey. What do you recommend to encode as data set? So for me, the encoding would be the unique samples for data set. And again, there's going to be variations depending what kind of experiment, if you're recording the same sample over and over. So that's the example I would have, the very simple example. But if you have more examples that would not fit in that frame, you could either ping me on ImageSC and ask me how you would, how I should recommend to do that, for example. Okay. Now, if I'm doing that of creating one data set each time I'm recording a sample and I'm having a lot of conditions or I do a lot of replicates, this make this list quite long. Okay. And that's maybe not ideal for most of us. So we're kind of used to have this foldering of data sets. So OMERO does not show here the data set with these folders. So we have only this projects object that is a whole project. And inside putting data set, and directly in the data set we find images. But I'm not able to put a data set inside a data set. It's not folders like we have on our computer. Still, people want to be able to organize in more levels, and that makes sense. Like it's useful to organize your data in the different conditions, or if you use different genotype maybe for your samples. Still, anyway, here I would always organize my data sets in this kind of flat list. But what I can use then to organize my data sets are not these projects, but what I can do is tag them. And so creating tags, I would then assign categories to my different data sets. So to put again some context here, this is a master module. And so we had students going to the microscope, learning about techniques. And we thought, how would the student like to find their data? And so we used the group of students. So the students were split into 4 groups. They were learning, working on some of our microscopes. So maybe the students also want to find the images that belong to certain microscope. They were learning about some techniques and a certain day in the module. And now instead of sorting my data set in this project list, what I would be able to do is to go to this tags tab, and now list the data set by those tags. So if I go to the groups, these are the 4 groups of students, and I could go to group 1 and find only the data set of group 1. Or instead, if I'm interested in finding all the data set that were matching this confocal lesson, the basics of confocal, this would be the data sets of the 4 groups. And this is kind of like making a folder. So I create these tags here. So creating tag sets, which are categories of tag. And inside each category, I would create my different tags. So group 1, group 2, group 3, group 4. And those are, yes, kind of folders. So here I'm only looking at the data set inside this folder. Now, if we want to do this kind of subfolders, so having more than single folders or single condition, or which one should I use? There is this extension to OMERO Web that can be installed that is called Tag Search. And in Tag Search, that would exactly like browsing inside your folders. And so I could type group 1 and I would move inside the folder group 1. These are all the data sets of group 1. And I can type a second tag, and from the list available here, these are only the tags that are on these data sets. And so I could say, show me the data set of the LSM880. And so this is the only data set of group 1 that was acquired on the LSM880. But the advantage of a tag is that it's much more flexible than folders. What I can do is first browse by the LSM880. So I first advance in that folder, and only then do I want to start filtering for group 4. So now we're kind of removing the conflict of which should be the higher level folder for our data sets. Maybe different people disagree in the lab. Here, tagging your data in that way, anyone could search in the order that's the easiest for them. And so for your tags, I would only recommend to use what's useful for you to sort, to filter your data. So the tags, I would not recommend them to use to add all the information that is about your images. So don't use for every fluorophore you've been using and magnification and etc. Because for most of the tags, you would never use as a filter. You would only use as an information that you need to know once you found the data set. And this is information that is already displayed in these key value pairs. So we will use then the key value pairs to start describing our data. So here going in detail. And what's the best way to bulk tag images? Again, a very good question from Ved. So for bulk tagging, if you want to tag your data sets, so right now this is the data sets that I'm tagging, because in the tags, this is the data set I want to find. I could, with pressing the shift key on my keyboard, I can select multiple data set, or I could also, with a control key, select individual data sets and go tag them this way. So that would be already tagging by bulk, like more than one. But if I want to do something more complicated, maybe I have hundreds of data sets to tag. We have annotation scripts for that. And I'm going to present them in a second, that we have a way to import tags and attach them to data sets by doing that bulk. And I have another question from Sergey. Are tags group one, group two added per data set or image by image? Yes. And so my tags are added on the data set level. So the difference here, putting the tag on my data set and I put this group two, I could also take this image and let's also add the tag group number two. Let's browse that image. And now you can see that under the tag group 2, I see my data set. That was, I think, this one. And if I see the data set, I also see the images that are inside. But I find the same image also under the tag. And my style to organize data set, I really recommend to tag data sets because when we see images, we want to know from which data set they come from. Or that's at least my feeling. But there's still use cases tagging images. So to give you some examples, and that's also what tags can be very good for. They're very flexible at annotating and organizing data. Um, so let's remove the tag from that image first, that I'm not messing up too much. But say I'm going to have a meeting with someone and I'm preparing ahead the meeting, what image need to be discussed. Maybe there's some problem we need to talk about in the image or how to refine our acquisition settings or sample prep. So I could go find the different images that were problematic, go to the tags and add the tag, maybe creating a new one to discuss. Adding this tag on the images, now I have a way to filter only these 4 images. And so I could go and say, for the next lab meeting, please have a look. I could also send a link to that tag that someone would be able to see the 4 image we're talking about. So I don't need to say there's one in data set here, another one in this data set. I can tag myself. Let's discuss these images. Discussion is over. We said everything we had to say about them. I could simply delete my tag. So I'm careful not to delete my images here, but I'm simply deleting my tag. This is gone. This is not cluttering my server with useless kind of organization. But when I did that, my image never moved out of the data set, but I was able to filter them. That would be one example of tagging images. Another example I have, if you are creating a training set, validating set, testing set for training artificial intelligence models, for example, and you would like to randomly select images and you don't want to select the same image twice, you want to know if it was in a certain set, etc. You could also do the same, selecting randomly images and assign a tag for training. So those images would then be part of the training set for my model. And those other images, oh, one is part of training already. Yes, something like that maybe. And they would be part of the testing set. So that my tags are creating folders inside my data sets. The data set for me is really my image library, my data library. That is the very organized collection, all in a flat hierarchy, while my tags are really the way I need to think on how I organize my data, that I can have different ways of filtering. Okay. I think that's pretty much I had to say about tags. So now let's dive into this other kind of annotation, which are the key value pairs. And we'll come to talk about bulk tagging. So for these annotations, we are following the RMB guidelines. So this is a publication in Nature Communication, I think, or Nature Methods. That is describing what kind of information we should record for our microscopy images when we are publishing them, that someone else could understand. These guidelines are not, how to say, very explicit on how we should write it. So they're really saying this is the kind of field you should record. Try to call it like that. But because biology is biology and all projects are very different, and some of these fields do not really fit our images, or our projects have certain fields we would like to record that are not generic enough to be part of the recommended key value pairs, recommended annotation, sorry. What we are doing in our facility is to having a discussion for the different projects on making those templates. So we are doing this discussion to try to push people to use that more. This is still something you could do on your own. What kind of template you would like to have for your as information. So making your templates of metadata. And the thing here in OMERO, so you can type your key value pairs here. So saying this is property A, and that's going to be the value A, property B, and that's going to be value B. So this, you could very well type yourself one by one. For the first data set, you're going to have in a way or another to type this information. But once you have it for your first experiment and say that your data sets are can either replicate, so the information you're recording on one data set, you should record the same information on the next data set. So today I'm going to the microscope with my 3 new data sets. So let's go ahead and find the data of today. Sample 1. I'm going to copy that. That's sample 2. And that's sample 3. So these are my 3 new data sets with images. Well, right now they don't contain anything. This is really for the example. But the annotation I just made earlier that I'm not going to find here. This is something I could copy and paste on my next data sets. So all you have to do now when you're at the microscope, maybe waiting that your Z-stack is acquired, you could directly create data sets in OMERO, waiting for your images to be imported in those, and copy-paste annotation. This is not value B anymore for sample 2. This was value C. And so reusing my kind of template just by copy-pasting from one data set to the next. That works when I have a few data sets at a time. That is, I really recommend to do that by hand overall. So I'm generally not in favor of automatizing everything, because with automatic things, there's always the scary part that something goes wrong and I don't see it went wrong because it did not tell me there was an error. So overall, if I have the capacity to do this copy pasting by hand and checking one by one, that's what I would do. But if you have a lot of data sets, you're going to need to do some kind of bulk annotation. And so for that example, I'm going to take, again, this screen data set that I have here. So the screen data set, this comes from a 96 well plate. Well, in that case, we only acquired images in those few wells. So not so much. I could still kind of do it by hand, but that's at the threshold between at the threshold of being just too much for me to do by hand. I already have my template here on this well. So again, this is something I could copy and paste on the other wells and fix it one well at a time. But I mentioned earlier that we have these annotation scripts. So these annotation scripts have existed for a while in OMERO. I reworked them, I think that was last year, to work a bit more flexible, that they are a bit more flexible on how to annotate. And so what you could do, you could take annotation on a given well, and that I could export to a CSV file. So if I decided to export that for this well export, I could also include tags here if I wanted to. Right now, I'm just going to export the key value pairs. This is generating this file that I could open with Excel, and that's how it would look like. So right now it says this is the object ID it came from. So that's my well right here, 66054, 66054. This is for the well name C4. And here the first row would be defining um, all the keys in my key value pair, and the next rows would be for every row one individual object that I could, of course, then change the values here. So I could start copy pasting those, say, and use Excel in smart ways because, well, Excel can be used very efficiently to annotate a lot of wells at the same time. That's maybe some information you would already have if you were doing 1,000 experiments. So some kind of Excel file with information written. Here you would have to format it kind of in a way that OMERO would accept it. In the case of high content screening, so that would be we could do here by exporting using the object names. I'm gonna directly reuse, for the sake of time, a template that was already filled with information for these different wells. So that's the correct information here. Sorry. So with the information for the different wells, for the test, I'm also gonna add a tag, and that's gonna be low med high for the concentration, and let's call that very high, that I generated a column named tag. And this should be interpreted then in my script when I import this back. So using this file, I can, again, go select the whole plate that I would import from a CSV file, my annotations. So for this given plate, I want to be targeting annotation onto the wells of that plate, and I can choose my CSV file. Yep. So that would be my CSV file right here. And I'm going to import the tags. I allow the tag creation, so that's some safety measures that I would not create tags if I made a mistake in the way I was annotating. But the rest should be fine. That I'm importing my annotation and my tags. So if you're, well, let's finish that first. Let's go check if it worked. We have our tags for the wells, and we have the new annotations now. So this is something then you could use that if you wanted to bulk annotate data sets. And maybe another advice on how would I get all the names of the rows that I want to annotate or all the object IDs. You could select all the data sets, go to export to CSV, and here I would select all the data set to be exported. And if I don't want to export the currently existing key value pairs, there's this thing called namespace. I don't want to dive too much into that, but if I type gibberish, that would not match any of those key value pairs. That I could run this and download that. And now what I have here are all my data sets with all the object ID, and all I would have to do is adding a tag tab and starting typing all the tags I want. Okay. This was a very quick introduction as well to those annotation scripts. But if you are willing to learn more about them or how to use them, there is in the OMERO guides, read the doc. Walkthrough example, where would that be? I don't remember. Server-side scripts. And there is pages on the annotation scripts. So let me drop that in the chat. When I find it. Here. And there you would have some walkthrough example explaining again what I explained you just now. Okay, so we have. So where are they available? So if you have the latest version of OMERO server installed, they should come. They should be installed by default. Otherwise, you would find them in the GitHub repository called OMERO. Oh, sorry, scripts. And this is where you can find all the default scripts of OMERO. So the annotation scripts are part of the default OMERO now. And if you have again this kind of questions later after the talk, please come to ImageSC and we will help you there. Okay. So now I really took some time explaining this annotation, and I'm kind of documenting here my data. That can always be useful, of course, for the future when I'm coming back to my experiments and I want to know what's there. But is there really another value to that? So we were seeing OMERO figure before, and in OMERO figure, we also have the possibility to add this information on top of our image. So if I select that panel, for example, and I go again to labels, there is something called key value pairs. And so I could select that, add information, and here it would propose me the different keys of that image that I could show, for example, the content of channel 1. Well, that's DAPI, maybe not the content, but the biological entity, that is DNA nucleus. And so here we're saying we are also staining DNA nucleus because this is part of the things I recorded in my key value pairs. So again, biological entity, well, I guess that will not work by default here. But the idea is that if you have this condition recorded as your key value pairs, that you would also have a way to reuse them uh, in other places in OMERO, so OMERO figure. But if I take here this example of my plate, so I could, for example, select these 4 wells. In every well, we have multiple images recorded, so I'm going to select random ones around, and I can take the link to those. Let's create a new figure where I'm going to add my different images. Let's resize them, and we could directly show on the left vertical the key value pairs that would tell me what is the treatment of volume per well. No. What am I looking for? Dye concentration in channel 2. Dye concentration channel 2, like that. Okay, why did I put 2 of them? I'm not sure. Not sure what's going on here. Sorry. Okay, there's certainly an issue here, but let's pretend we're not seeing that. And let's align our images. And so again, I could prepare my figure quite rapidly, displaying one channel at a time that I could then create this kind of kind of figure rapidly and populating with metadata coming from my key value pairs. And so that would be another place where key value pairs becomes useful, that you took the time annotating your data, now that you also want to reuse it in other places. Okay. Um, so, where are we now? Okay. And now I'm going to move to the last part of this session. So we talked about annotations, and we were working with OMERO from the web interface. We saw some of these plugins that come with OMERO Web, so the figure, the tag search, the viewer, um, and really what here OMERO server is doing is giving us access to this image data that we can display it and interact it in some ways in the web. But OMERO Web for bioimage, sorry, OMERO for a bioimage analyst, there might be other ways that we could access the data so that we are not, again, using this graphical interface to access the data, because that might be a bit too slow if we want to be processing thousands of images. So the OMERO server also provides APIs, so application programmer interface, that we could access to that to the data from Fiji, from QPass, or also from Python. And I want to show you some examples on how we access this data. Though for the Python part, well, I'm again not going to go in details because we are not going to have the time, but there was also a session by Eric Ratamero, I think that was yesterday, on Easy OMERO, so how to access the data from Python script. So again, that's going to be part of the recorded lecture. And a lecture I forgot to mention while I'm thinking about it. When we were talking about metadata, there was also another session from Caroline Miller on Monday on Make Metadata Cool. So if you also want to hear more about annotation and Ruby templates, etc., you could also go see her session later. But coming back to our APIs and accessing data, with other ways. So I have right here my Fiji windows, my Fiji, and I installed in my Fiji the OMERO plugins. So I could from OMERO connect already here to OMERO. You are not seeing on the side, but I have a login window, so I'm typing my password. And here it's coming. There. And what it shows me here is another graphical interface from Fiji that I could also go and access my image data from there. So I could access this data and open it. That would directly open inside Fiji. And like that, I could open my image in Fiji and do some image processing. So maybe detecting nuclei. I'm not going to write an image analysis pipeline right now, but I'm going to create some ROIs, or I could also say, let's make ROIs around those cells. And this I could save as a result to OMERO. So save region of interest to OMERO. I'm going to save it for the current window, saving this ROIs like so. And now if I go find my image again, well, I'm going to have actually a lot of region of interest. There were already some region of interest before from some image analysis we did on the nuclei. But here you could see the rectangles I just drew in Fiji. And so that's how you can start connecting image analysis to OMERO. So OMERO is not an image analysis software. It's really here to help you organize or serve data. And so accessing the data from Fiji, I would do my image analysis in Fiji and save my results to OMERO. The nice thing about that is that I could also share a link to those region of interest I just created. So someone else could review them, or I could send a very specific portion of my image to one of my colleagues that they could also have a look directly, that I do not need to describe in my email which part of the image they need to look at. But here we're still very, well, we're still doing all this manually. So I had to open my image, double clicking on the graphical interface here, and I had to go save my ROI manually. But in Fiji, there's also the macro language that exists, and there are ways that you can write macros in Fiji that you would access to the data. So the plugin I'm using here for Fiji is the OMERO macro extension from Pierre Pouchin that wrote this. So you would find also on the GitHub. I can also give a link to that. You would find on the GitHub how to install and also some information on how to use. But the best way to start with this, if you go to the templates, you find actually examples of how to write macros in Fiji, that you would have the macro extension. And here's an example that would connect to my server. So all the first part here is connecting to my server. Here it would be listing all the images inside the data sets. And for every image in that data set, this is where we are applying our macro, so doing a threshold and detecting the nuclei from that threshold and saving ROIs. So now I could have 100 images in my data set, or I could also have a list of data set that I could iterate as well and process the whole data. And I have a question from that. In Fiji, when you fetch OMERO images, do they get downloaded on the local machine? So the answer is yes, but maybe there's different way you can think of download it. So it's not creating a file on the local machine. It's really just loading it into the memory of Fiji. So when I open it, yeah, I create this image. It's loaded inside the memory. Well, there's also the possibility of doing it with this virtual stack. But the image is not saved on the computer. So you would be loading, fetching the image data when you're working with it, but not saving it locally. There could be use case that you need to save it on the machine to sort of create a buffer or have faster access if you need to reopen the same image over and over. So it would make sense in your macro that you download the image, save it, and then close it, reopen the one image if you need faster access. Okay, and this would be the style on how to write a macro to connect to OMERO in Fiji. So again, sorry for that, going a bit fast here, but I really want to give you some clues on how you could automatize processing of data on OMERO from different image analysis tool. And again, here, if it does not really, you think it's not really for you to write macros, maybe it's the case for someone else in your lab. And maybe someone else in your lab is an expert in doing image analysis, or someone in a microscopy or image analysis platform. And that brings me back to my points of annotating data, that you would have to do really, you would have to do your annotation job carefully, that you are putting all the necessary information on your data set, that someone else could do this kind of macros in case they need information from your images to be able to process them. And to show again in this macro that in my templates, there is also something to read key value pairs. So maybe I'm putting in my key value pairs a parameter that's important in my image analysis. So so that I would then use that information in my Fiji macro. And what we're doing here in Fiji, we can also do it in Python. And so I have a small demo here that I am connecting, well, importing the libraries required to connect, giving my username, password, and server address, to connect to the server. So here it shows me that I can connect. The next thing I'm doing, for a given annotation ID, I want to list the data sets. So this annotation ID is coming from my tags. So tags are annotations, and I could say, give me the list of data sets here that have this tag ID. So let's take 95105, 107, sorry. I need to hide this. uh, And so 95107, this would list the IDs of my data sets that have this tag. And because we are programming here, again, what we could do is that for every data set that have this annotation ID, so that's the ID we just listed before, we could list all the images inside each of these data sets. And that gives me this result. So here I'm really showing that I can list my data, my images from a tag. And here I could think of any kind of logic of combining tags, so doing this kind of intersection of image IDs that I would only work on the image IDs I have to work on. Or I could also create a tag just for the purpose of processing certain images that I can do my filtering by hand before and process only the data sets I have to process. And to finish this example in Python, so I just took the first image that I would connect again to my server to get the image, so getting the pixel values of that image. Here I'm displaying the shape, so we have one Z slice, one time points, 460 pixels in X and Y, and 4 channels. And I'm using then this matplotlib library in Python to display the content of my image pixels. Okay, and just to be complete and compare that example with Fiji, I could also using this EasyOMERO library, getting the annotation ID of these key value pairs for a given data set and getting the content, so that would give it to me in this dictionary structure. So again, if I need some parameters in my analysis, that is something I could use here. Um, okay. What did I wanted to say? Um, yeah, I think that's pretty much everything I wanted to cover here. So I hope um, this gave you some convincing argument on why you should use OMERO to annotate your data, or the list annotate your data, that adding metadata might be important to reuse in different places. What I find useful about OMERO is that there is already this existing database that when I write image data in OMERO, it is organized, it is consistent, and I can use then my image analysis scripting to go process this data and access the data that is organized in this database. So any information I'm adding is going to be useful for me for my processing. And so let's go finish with the slides, trying to recapitulate a bit everything we saw in this session. So here is this logo is the database of OMERO, and me as a researcher, I'm creating image files. Putting those files in my OMERO database, what I get are images, and those image pixels that I can go and review and collaborate with my teammates to work on those. Adding more images only make the whole thing grow bigger. I'm organizing my image in data sets, projects. I'm adding annotations to add information. So growing, and this is the work done either by me or my PI, that we could collaboratively annotate our data. Then someone else would come, do image analysis, so pull my images, maybe also pull annotation metadata, create measurements or a region of interest that they would then push back to the database and adding information. Once this is done, all the data that is in my OMERO, I could again extract it, most likely with scripts here to create this kind of data frames. If you don't know what a data frame is, you could think basically of big Excel table where every row is a kind of measurement. And you would have a lot of column with all the properties. So all the property you define with your key value pairs, explaining what were the different conditions, or all sorts of measurement you made, for example, number of ROI per image, or size of the different ROIs, or the content of the pixel values inside your ROIs. So really generating these tables that you would then create, well, extract statistics from it and do different kind of display to create your other figures for your papers. And so that's yeah, that's basically the idea of how you organize data and do data science on your images. Other resources I could point you to. The i3D Bio projects also aims to push forward data management, mainly in Germany. We produced training videos, so that's also something you could check out if you want more. Overall, I would say you should go probably on YouTube. There's going to be other videos on OMERO that would explain. You would find a lot of resources online. If you feel overwhelmed by the quantity of either information or options you have and you don't really know where to start, I could then only recommend you to come to the forum image.sc, ask your question, and you would get the opinions from different experts there. So really to conclude, data management is something that needs to be taken by everyone in the lab, from group leaders to the analysts. And really that's the purpose of data management for you in the lab is to increase, the purpose is to increase your ability to exploit data. You have remote access to the data to share links over files. You can have this traceability from the acquisition to analysis. And then the people working on OMERO can work on extensions to solve specific needs. So because there's this database filled with images and annotation, then people can contribute some plugins or work on that would help you work with your data later. Okay, and this would conclude this session. So I will, well, we are really finishing exactly on time, but if you have some question, I'm happy to stay a bit more to answer your question. Thank you for your attention.